Hey Shane.
Hey, how are you?
Hello?
Yep, can you hear me?
Yeah, now I do.
How are you?
I'm good. How are you?
I'm good.
Um, can we go over the BB section of the test that I took that weekend?
Mhm. Yeah, I think we finished Chem Phys, right?
Um, Wrong tab. Hold on. If we're doing a longer lesson, we just go over all of the problems because I feel like last time we didn't do the ones that I flagged.
I think we did
We didn't do 28.
because I wrote down which ones were flagged.
Yeah,
Maybe,
but we never went over them because I just clicked incorrect answers
yeah,
during a review.
I wrote down which ones you flagged, so we can look at
I feel like it's not like I have a pretty good understanding. I really want to do VP because my score here isn't great either. So I feel like we're really focusing too much on physics.
yeah. So why don't you do like filter flagged, and I can write them down. All the way on the right has a flag. Yep. Okay, 4, 17, 21, 25, 26, 40, and 47. And it seems like, so 26 and 47, I'm circling so that now when we do the um, incorrect ones, we can just add number 26 and number 47 to that.
Uh-huh. Okay.
Hit the filter button because I don't think that, yeah. Okay.
So butane is 5, right?
Nope,
But 3?
nope.
And I don't know what it is.
So maybe the choices could possibly help. Or they're also saying a pyrimidine dimer, so that could perhaps help. But
I mean, honestly, the thing is when my class did like dimers, we only really went over like the damage that a thymine dimer could have. Like,
okay,
what it means for DNA. So,
yeah. But you took organic chemistry, right? Right.
like, yeah, but no.
So like, yeah, one of like the first things they teach you in Orgo is the like the names for the different number of like carbons, right?
Mm-hmm.
So like, okay, so like meth would be how many carbons,
1.
right? What's what about if there's two carbons? What is it called then?
Well, I'm thinking like methyl ethyl.
Oh, which one?
Methyl is 1, ethyl is 2.
All right. So keep going.
Isn't it T like tri or tetra?
Um, so next is going to be for the three, it's pro.
Propane. butane. So there's butane 4.
Yep.
And then 5 is pentane.
And 6 is
Hexane.
7.
Septane, octane.
Nope.
What is 7?
Hept.
Hept? Okay.
And then for 8.
Oct.
9.
And I don't know.
It'll be non.
Okay.
And then what about 10?
Deck.
Yep.
Okay. So butane is 4.
Mm-hmm.
Would that be like these? 4 well, that's not a carbon. So this.
Okay.
Pyrimidine.
Mm-hmm. What's
I
that? A pyrimidine? What's that?
don't remember.
You remember pyrimidine?
Mm-mm.
Try it. We'll try it out. Like, didn't you just take like a genetics class?
Yeah.
And they didn't teach you what a pyrimidine is?
No.
That's weird.
Why?
So pyrimidine is a type of like base. Like C, U, and T are all pyrimidines.
Oh, purines and pyrimidines.
Yeah.
Cut. Yes.
So you definitely want to know
I
butane
do.
names for that. You
Like
def
structure, I don't know like what it looks like.
uh-huh. Well, I mean, well, you didn't know what it was at all. I asked.
Cut pyramid. Well, I knew like, all right, I get it. It needs to come to my head.
Yeah. Especially, yeah, because if you're not understanding what like butane could look like, then yeah, it's not a good position to be in. And also if they're saying that this is a pyrimidine dimer, we can and you know that the difference between so I mean, what's the difference between a purine and a pyrimidine?
Um, Purines are AT and pyrimidines are
Oh,
C.
yeah, but I mean about the structure.
Um, they have 3 versus 2 rings,
Mm,
or not the rings. I don't know. I was taught
what are you talking?
weird way, like cut, pyramid.
Yeah, but I'm saying like what would they look like? Like if purines are different from pyrimidines, they should look different, right?
Yeah, but I'm not sure. Like
And so in our choices, we see what type of stuff.
we see like benzene, and well, not we just see like rings.
Okay. And go from there.
Assuming that pyrimidines would have like extra bonds.
So you
Maybe.
see that there's certain things with 2 rings, certain things with 1 ring.
Like how this has. I mean by 1 ring.
So what were you saying that has?
There's an extra pentane. I don't know. It's like a 5 carbon, or but there's
So how many rings is that? Would that have?
3.
Nope.
Does this stop being a ring because there's something bonded to it?
No. It's just that the cyclobutane is what connects the pyrimidines in the pyrimidine dimer. So you're not going to count the cyclohexane when you're determining if something's a pyrimidine or a purine. And so if you're not counting the cyclobutane, tell me what you are counting.
The other one?
Uh-huh. So okay, so the cyclobutane is the thing that connects the pyrimidines in the dimer, right?
But I don't know what's the dimer here.
Well, what would a dimer be like in general?
Like UV rays cause like
But what is the dimer?
more dimers.
You're telling me what
Right?
dimers, like how they're like you're telling me that UV light can cause dimers, but you're not telling me what a dimer is. Like, what is a dimer?
Like a kink?
No,
I have no idea.
just look at the word.
Dime 2.
Uh-huh. Like, what if it was just one? What would it be called?
Monomer.
Uh-huh. So dimer would mean.
Like 2.
Mhm. So now go from there.
Right.
So we can see that the cyclobutane is what connects the 2 monomers to form the dimer.
Like I'm confused. Which one would be a dimer?
Well, all of these are dimers, aren't they?
How?
Well,
Oh, they all have to. Wait, why?
so why don't you try to tell me why?
So there's no like specific structure to a dimer, it's just something that connects.
Well, I mean, there are specific structures, but since you don't know what a pyrimidine looks like, we're gonna try to figure it out by just looking at the choices, and we see that the cyclobutane is what connects the 2 things, the 2 monomers for the dimer. So you can now just look at the choices and you can see what's to the left of the cyclobutane, what's the right of the cyclobutane, because those will be the 2 things that combine to make the dimer.
I think like the 2 halves are connected by the cyclobutane.
Uh, well, yeah, but like, so look at what the difference is for the one on the left, one on the right.
So do they have to be identical on both sides in something like C?
So before that, You what I
Oh, well, these aren't identical.
so
Like,
that's what I asked. So when you're on D, what were you seeing?
I was saying that there's like an extra like ring over here.
Okay. So it's connecting something with two rings to something with one ring, right?
Yeah.
Okay. And so now what do you think pyrimidine and purines are? Like how they differ? Because before you said one has two rings and one has three rings.
Not rings, bonds.
Well, I mean, they have a lot more than two and three bonds, but I'm saying for the rings, like maybe that has something to do with it, which is why for all of these you see rings, right? And then sometimes you see two rings, sometimes you see one ring. So maybe that has something to do with it. So, okay. So a single ring will be for pyrimidines and a double ring,
Is for purines.
yeah.
So this would be attaching a pyrimidine and a purine. So they do have to be the same on both sides,
Mhm.
right? Or is this fine? Like, because there's two, would these both be purines and this would be fine if it said pyrimidine purine? Oh, sorry, purine dimer.
Uh-huh.
So it's C.
Yeah.
But so even though it's like NH2 and O, it's still fine.
Well, I mean, those are purines, though.
Right, but I'm saying if it said cyclobutene purine dimer, would B be correct, even though there's like different bonds here?
I guess it could be a purine dimer, but it won't be a cyclobutane one because we have nitrogens part of the
Right, because it's
form
not
of the ring.
carbons. Okay.
But yeah, you definitely need to know the naming conventions here. You definitely need to know what a pyrimidine and a purine is, and you definitely need to know what a dimer is.
Okay, so I need to know the naming conventions. Meth is one, like eth is two, bute is three,
Nope.
no, prop is three,
Mhm.
bute is four, pent is five,
Mhm.
hex is six, hept is seven, oct, non-dec. Pyrimidines are CUT
Mhm.
and to have one ring, and purines are what, A and G, and have two rings.
Mhm.
And a dimer, it's like monomer dimer. So we know that it has to have, well, the whole thing is a dimer. It connects the connects the two
Mhm.
ring structures.
Yep.
And that's what makes it a dimer, is that there's something connecting them.
Yep.
Okay.
So even if you didn't know these, you can tell that, okay, A has a single ring on the left, a double ring on the right. So they can't both be pyrimidines, so that's out. B has two rings on the left, two rings on the right. That could work if a pyrimidine was two rings,
Uh-huh.
and then C, one ring on both sides. So that could work as well. And then D, one ring and then two rings. So D is out also. And then between B and C, you can then, if you didn't remember what the pyrimidine, like whether it's two rings or one ring, the cyclobutane can help you because we have the nitrogens,
Right.
there, but we have our cyclobutane there.
I'm curious what I picked to begin with. What? It doesn't show.
A, pick B.
B. Hmm. Okay.
Yeah.
Aluminum phosphide. So it gets oxidized. And the hydrogens get reduced. So if it's more stable oxidized, then it likes a higher pH.
So what is this question asking?
AIP exposed to an aqueous solution in which pH range will result in the largest amount of phosphine production. So it, like I said, it gets oxidized,
So the question's asking us about pH range, right?
right? And phosphine is like the result. That's the aluminum and pH.
So wait, so the question's asking us about the effect of pH.
On the production of phosphine. I understand that. I'm just
So
to think of what aluminum phosphide is like an acid, right?
um no, but something else could be one. Like what's happening in reaction one?
It's getting oxidized to aluminum 3.
So what's being added to the ALP?
3 hydrogens.
Just 3 hydrogens or
Yeah.
3 protons, right?
Oh, well, yeah, but
Which means,
it's becoming more basic
well, so the protons must come from,
somewhere else.
from like an acid, right?
So it's getting denatured. Uh,
No, just if you
how
just
do
reaction, what's happening?
we're mixing AIP with the 3 protons.
Uh-huh.
We're getting aluminum 3 plus and pH 3.
Okay. So now
The phosphine.
what pH range will result in the largest amount of phosphine production?
Well, it's not an acid, so this one's wrong.
Why?
I know you're saying that it's getting the protons. So you're saying if if it was an acidic, it would lose more protons.
So this
And
is saying that, so these H pluses can only come from an acid, right?
right. And so that produces 3 phosphines. But if there were more protons, then there would be more phosphine.
It's not making three phosphines,
It has to lose.
but it's not making 3 phosphines, but yeah, if you increase this reactant, it would drive the reaction forward, right?
So lowering the pH
Mhm.
would create more protons.
Which
Or
would mean more of the reactant, which means that it'll be shifted to the forward.
see, I was going about it like in the completely wrong way. I was looking at it and I was like, this is getting oxidized, this is getting reduced. If it's getting oxidized, it's
Do you want to see that? So that's why I asked you to look at the question,
right, I know.
because the question's asking about which pH range will result in the largest amount of this being produced.
But isn't like the way that I thought about it, even though I didn't get the right answer, if it's losing electrons,
So
oh, because then I would have picked a positive.
here
Like,
you
okay.
you see that they're asking about a pH range, right? And if
Okay.
you're asking about that, we know that pH affects
The
phosphate.
reaction.
So when you look back at reaction 1 and you know that you're looking for pH range, the only thing that's relevant to that is the protons,
Mm-hmm.
right? So pH range, thinking about like, is this going to be acidic? Is this going to be basic? So when you look back at reaction 1, even though you thought it was redox first, after looking at the question, which asks you about the pH range, when you look back at reaction 1, it should then be apparent to you that this has something directly to do with pH.
Okay.
And then we wanted to do, because you also were going to eliminate the right answer here. So you don't want to do that. You want to think about the question stem and how that relates to the reaction that we see here, and then what pH range would produce the most phosphine.
Okay. Can you explain feedback inhibition to me before?
It just means, the more, let's say, product you create, it would create a negative feedback loop, giving a signal to the thing that's making that product to tell it to slow down in making that product because you have enough. So that would be negative feedback or feedback inhibition.
Mm-hmm. Wouldn't that be what's going on here, though?
Possibly.
But it's allosteric regulation too, because there need to be stable. This is Krebs cycle, right? Or glucose?
What do you think?
Well, I know this is in the Krebs, like it's one of the steps. Like I'm thinking, no, glucose, Krebs is the long one.
Yeah.
I think the answer is C.
Yeah, because so this happens in glycolysis, and the phosphofructokinase step is the second irreversible step of glycolysis, which involves taking fructose 6-phosphate and phosphorylating it into fructose 1,6 bisphosphate,
Mm-hmm.
which means that is this up here, like what I'm going to write here, this could be either ADP or ATP. Which do you think it'll be?
ADP.
Um,
Isn't ATP used in order to break this down?
well, what's happening from this to this?
Isn't the carbon that it's bonded to changing?
So what is this?
Fructose 6 phosphate,
What's
or
this?
1,6 bisphosphate.
So what's happening?
2 phosphates now.
Uh-huh.
The bond between fructose and
So we're going from 1-phosphate to 2.
yes.
So
So it'd be ADP to triphos. Okay, yeah. So it's ADP to ATP.
no. So what's happening to the fructose 6-phosphate?
The fructose stays the same, and now it goes from phosphate to bisphosphate.
So what does that mean?
Doesn't this break the bond between the fructose and this phosphate?
No. So okay. What is this called again?
Fructose 1,6 bisphosphate.
So what could that mean? Like, how is it different from just fructose 6-phosphate?
Structurally different. There's two phosphates.
I'm asking, yeah. So if there's how many phosphates does this have?
One,
How many does this have?
two.
All right.
So it's gaining a phosphate. How could it not be ADP to ATP if it's diphosphate to triphosphate?
If it's ADP to ATP, the phosphate is going to the ADP to make ATP.
And this is diphosphate, so you're taking away this phosphate.
What? So if you're going from one phosphate to two phosphates, are you taking away phosphates?
No, you're adding one.
All right.
So
So
does that phosphate come from here?
how could you add it to both the fructose 1,6 bisphosphate and to ADP? So how many phosphates does ADP have?
Two,
What about ATP?
three.
And this has one phosphate and now it has two. So you're saying that we go from how can we go from fructose 6-phosphate, which has one phosphate, to the one fructose 1,6 bisphosphate that has two phosphates? We must be adding a phosphate group, right?
Mm-hmm.
So how can we add a phosphate group to both get this product and to get ATP?
You can't.
Yep, we can.
But if you're adding two, then you can't use ATP either.
No. How many are we adding?
Literally says two.
From?
Oh, but it's from one. Okay, I get it. I need to switch to in the computer. I understand. So it's ATP to ADP.
So tell me why.
Hold on 1 sec. Because you're going from fructose like fructose 6-phosphate, which has one phosphate, to fructose 1,6 bisphosphate, which has two. You need to add a phosphate to get that, while also you need to add to like the second the second one in ADP. See, that part I'm still confused about is like,
What are you saying? So
because it's not like it can like just I thought that ATP to ADP it only loses one. Like that's what
but how many is this gaining?
this is just gaining one. So then
So
that
this
means
would lose
ATP to
one.
ADP is just losing one phosphate because that phosphate from here went to make this.
Yep. So
Okay.
so this should this here be ADP?
No, so it's the opposite.
ATP to ADP, right?
Mm-hmm.
Because you have to ask yourself, if we're phosphorylating the fructose 6-phosphate, where is that coming from?
From the ATP.
Yeah.
But it's only losing one overall. That's what I got confused about because you were like, it needs to add this to this too, so it wouldn't work. But it's literally just in
We're adding
order for this to get to this, it needs to move one from ATP and make ADP.
yeah. But I'm saying that that's because so if the fructose 6-phosphate is gaining a phosphate, it must mean that something else is losing a phosphate. So if you said ADP to ATP, that means that both fructose 6-phosphate and ADP are gaining phosphates, which is impossible, right? Because where's the phosphates coming from?
Right.
So should be ATP to ADP.
Right. And that's
Mhm.
and because if one thing goes up, the other goes down, that's why it's feedback.
Oh no, it's just that so this
That's
enzyme
it.
happens to be inhibited by the production of ATP because ultimately, as glycolysis proceeds, it makes a net positive amount of ATP. And if you have a high amount of ATP, it means that it's gonna tell this glycolysis to kind of like slow down and not let this reaction proceed any further. So that's the feedback inhibition. If you have a high concentration of ATP in your, in your cells, that means that you've already, like the point of the glycolysis is to make that ATP. So if you have enough of that ATP, you don't need to do that glycolysis anymore. So that's why that would be a negative feedback or a feedback inhibition, meaning high amounts of ATP would bind allosterically to the phosphofructokinase 1, um, and have it do what it does less, essentially.
Okay.
Yeah.
Cool. Dendrotoxin from the mamba snake blocks voltage-gated potassium channels in the somatic motor neurons that regulate skeletal muscle contraction. Would you initiate with initial exposure to dendroxin affect the ability to propagate an electrical signal in response to stimulate?
So this could be a good
I said action potential.
so this could be a good opportunity to review the neuron, like,
Structure.
so yeah, like the action potential stuff.
Okay.
So let's see. Okay, so if we have a neuron here and outside is whatever, the extracellular environment, um, and inside of the neuron, you have a certain concentration of sodium ion, a certain concentration of potassium ion, and same thing in the extracellular space. So relatively speaking, which side, like inside the neuron or in the extracellular space, which side should have a higher concentration of sodium?
Inside.
Mm, so
Isn't it 2-3 ratio of sodium to potassium?
so what would the resting membrane potential be for the
Zero.
neuron?
Resting membrane potential 0.
No. So remember we went over this pretty recently? Maybe you can start by drawing stuff that can help you remember maybe.
I mean, I know we're talking about action potential, so I'd probably draw that.
Okay.
So we had like kind of thing.
Yeah.
And then I remember I had to like get over like this was the action potential, right?
It'll be
Yeah,
a bit
I
higher.
drew this wrong because this was more like lower.
Mhm.
So
So what happens? So what is this
It'd
line?
be like the 70 line, because it's -70, right? The
Huh?
lowest point.
So the resting potential is going to be -70 millivolts.
And then the top is 40.
Um, +40, right?
Yeah.
And so yeah, what happens first? Like what happens here?
Um, it's when like a
So you'll have
it starts the reaction, like a substrate.
so this dotted line is going to be the threshold. And so if you have so action potential or like a stimulus can cause um a change in voltage,
Mhm.
and it can, let's say, have like go up a little bit, but if it's
It doesn't pass the threshold, then the reaction doesn't start.
correct, but if it does pass the threshold,
It's depolarized, right?
yep, so depolarization means that we're going from a negative up to a positive, right?
And then once it reaches that, that's the action potential, and then it gets repolarized.
So now let's think about what that means, the depolarization. Like, how does that happen?
It loses electrons.
Uh, I mean, not really. Maybe something else. Look at the stuff that's here.
Um, is it at like resting when the concentrations are equal inside and outside the cell? And then
Wait, um,
if something comes in,
where is it normally?
-70.
So, what do you mean about it being equal? So, if we go from negative to something positive, what can happen?
I don't know.
I mean, just think. How would you go from a negative to a positive?
Like when it gets oxidized,
I mean, well,
that's when it loses electrons, gains protons.
that's not really what's happening here. Think about the inside of the neuron and then the extracellular space.
It's gaining protons,
What do you mean?
a positive charge.
But like, wait, what are you saying for that? Why protons?
Because you said no to losing electrons, but
But do you see us writing anything like writing protons?
no.
Okay. So if we're going from something that's negative to
Like
positive,
potassium to enter the cell. That's
so it'll be sodium. So there's a sodium channel that can open up, and there's high sodium concentration outside of the cell. So if this sodium channel opens, what will happen?
if it's high outside the cell, then the sodium wants to come in.
Uh-huh. So you'll get your sodium influx, right?
It makes potassium want to.
So when you're looking at this and you're trying to figure out if we're going from a negative to a positive, and we already wrote out positive cations here, like you want to actually use that instead of saying like, gains or loses electrons or gains like protons, because we didn't write anything about electrons or protons, right?
Mm-hmm.
But if we're writing things about positively charged ions, then that could just be the thing that comes in.
Okay.
So this up portion is depolarization.
That's sodium coming in.
Yep. And then it reaches some maximum point here.
And it gets repolarized, which I'm assuming is potassium going out.
Yep, because now we have, compared to the inside, low potassium. So if we open up a potassium channel, the potassium should be an efflux, meaning it's exiting the cell.
Okay.
And so it'll go back down to the negatives, but since the potassium channel closes very slowly, it can overshoot it a little bit, which would be hyperpolarization, but it'll eventually go back to where it's supposed to go. Right?
It's the like divot thing.
Uh, like, so this part is hyperpolarization,
Like,
and then yeah, it'll eventually go back to the regular resting membrane potential. Now, the resting membrane potential is kept at a negative because, I mean, it, should be at equilibrium. It should have a charge of 0, but that's what the sodium-potassium pump does. Sodium-potassium pump makes it so that this cannot reach equilibrium because you're pumping sodiums out, like 3 sodiums out and 2 potassiums in, which means the pump is making it have a net negative charge through the use of ATP. And that's what keeps the resting membrane potential at the negative value.
okay.
So now tell me, like, explain it to me now about what happens here.
Okay, so we have like the resting potential of negative 70 millivolts.
And why is
And
it at that?
huh?
And why is it at that value?
Because that's below the threshold.
Well, I mean, no. So like, why is the resting potential negative like that?
It just is.
So I just talked about it.
That's below the hyperpolar. That's the polarization level. It's below the above the
Why is
hyper.
it though? Why is it a negative value?
Can you explain it one more time?
Yeah, so it's because the sodium-potassium pump. The sodium-potassium pump moves 3 sodiums out,
And two potassiums in.
potassiums in, and it does this against the concentration gradient, which is why it has to be using, ATP, right?
Mhm.
So yeah, so why is the resting membrane potential a negative?
Because of the sodium potassium channel, because it goes against the concentration gradient.
So these are the channels,
Right.
but this is the pump.
Okay, so but it's the pump, not
Mm-hmm. So what would the difference?
doesn't the channel like do the like three or no? Okay, never mind. These just like okay. So you get two sodiums that leave, and that's the depolarization.
3
Be
sodium?
Sorry, three. And then it reaches the action potential.
No. Um okay, so
That's
the
the
pump is what does the pump do? Sodium potassium pump.
the pump takes three sodiums out and puts two potassiums in, and in order to do that, it uses ATP because it goes against the concentration gradient. So it's active transport.
So and that uh results in what? Like what does this sodium potassium pump like end up doing?
A negative.
So tell me more.
And I don't know, it's just always been drawn that way.
Well, but you drew it like so you can't answer something by saying that's just what it is. You know, you have to I'm trying to ask like what's this doing, right? And you can't just say that's just how it is because first of all, when you first drew it, you drew it wrong. So it's not helping you to just say that's how it's drawn because you draw it wrong. So you want to think about what the actual purpose of all of this stuff is. Um, instead of just saying that's just how it is. So, and this is a high yield topic that you definitely have to know. And, you know, so why is the resting membrane potential at a negative value?
Because there's like it's off equilibrium now. There's
So the pump, right? The pump uses energy to have a net negative charge because it's removing 3 sodium plus ions and it's gaining 2 potassium plus ions, which means that it's going to have it's going to create a net charge that's negative. So the sodium potassium pump is what maintains that negative resting membrane potential, right? So that's what that pump does. And that's why it's kept at the negative 70 millivolts. Now, what happens if some stimulus causes the the voltage of the neuron to reach the threshold? What happens when that happens?
okay.
So what happens when that happens?
When it reaches a threshold, the reaction can start.
So go on, tell me what happens.
And so when that happens, that's when sodium enters the cell.
Mhm.
And then that's when it gets depolarized
Mhm.
and goes up to the positive charge, and it eventually reaches the action potential, which is 40 millivolts.
I mean, the action potential is kind of like the whole thing, but yeah, the sodium channel opens up, and since
Or
the
what is this? The like highest, the maximum?
what's that? Oh, the pump creates the concentration gradient, and that means high sodium outside, low potassium outside. And when that threshold value is reached, it'll open up the sodium channel. So a channel is different from a pump in that a, a pump is like using energy to move things against their concentration gradient, whereas a channel is just literally opening like a pathway that sodium can travel through. And if we open up that pathway, it'll travel through diffusion high to low. So you'll get a sodium influx. And if you're getting a sodium, which is positively charged, if you're getting a positively charged, cation entering into the cell, it's going to make that cell more positive, which means less negative. So that's why you see that depolarization bring the membrane potential up to this positive, like 40 millivolt region. And at that, when it reaches that, the sodium channel closes, but that's when the potassium channel opens up. And what happens from when that happens?
Then the potassium leaves, and that's the efflux.
Mm-hmm.
And that's when it gets repolarized.
Yep. So, and repolarize means
It
what?
goes back down to the threshold, but sometimes it can get hyperpolarized and go like too low, but then eventually it like
So,
stabilizes and goes back to
it's getting
goes
a more,
down. Yeah, it's getting more negative.
yep, because you're now getting rid of the positively charged potassium ions, right? And so it'll go back down, like it'll go past, like the threshold, because it'll have to go back to the um like this value here, which is the resting membrane potential. But yes, since the potassium channel closes slowly, it'll overshoot it and make it go result in a membrane potential that's less than the 70, like even lower. That's just the hyperpolarization, but then it goes back to normal.
Okay.
So yeah, next time we meet, I'll ask you exactly all of this stuff again. So I'm going to ask you what's the resting membrane potential, and why is that the resting membrane potential? And then I'll ask you the relative concentrations of sodium and potassium inside the cell and outside the cell. And then I'm going to ask you to tell me what happens for like each step of this action potential.
Okay.
So just
All
because
right.
you have to definitely know that. So yeah.
Andrew, okay, but now
Mhm.
if it blocks the potassium channels, then Na can still, like sodium still comes in
Mhm.
and the pump still works.
Okay.
It's just that potassium isn't leaving, so the repolarization can't happen.
Yep. Mhm.
So it would prolong the action potential.
Yep.
Okay. What is refract? Oh, but this is refraction like mirrors.
Oh, no, no. Refractory period is just the period like kind of between here and here before another action potential can happen.
Okay.
Uh
Uh,
yeah, because it's like there has to be some type of time between the next action potential.
and so it would prolong that too, because if this whole reaction is taking longer and there's a set amount of time, then the next reaction can't start.
Uh yeah, sure. That should work. And if you go back to that question and you hit the solution button, you should see a diagram. And we've seen this before, but and you probably saved it before, but this shows you everything that happens. Well, I guess it doesn't tell you about the resting membrane potential, but I would, screenshot this, and
How come this one says 30 and not 40?
it's going to be something in that range, so maybe,
Hold
well,
on, I want to take a picture of my phone because I feel like I have so many of these.
so it seems like the top is a little bit higher than the 30, but
Okay, let me take a picture.
the idea is the same, yeah.
So protein X. I think this part is what I used. Exhibits increased fluorescence. So sulfonic, wait, I don't think fluorescence upon binding to hydrophobic surface residues of proteins
So
is used for conformational changes.
what is the question asking?
Subunits of protein X are linked covalently by bonds between the
So subunits indicate what type of structure of protein?
secondary.
Nope.
Aren't the subunits alpha and beta?
I mean, they could be called those things, but uh, so that means that there's a quaternary structure. And they're asking
Folded protein.
how the subunits can be linked covalently. So what type of, I guess, interactions or bonds are made when, you go from like a primary structure to like a secondary structure, or like a secondary structure to a tertiary one? What type of bonds are made?
Disulfide.
Uh, so tell me, what else?
Went over that
What's up?
there's the amino acids or they form the peptide bonds.
So that would be the primary structure.
Yeah.
So what would be in like a
Cotton.
secondary structure?
Isn't it just a polypeptide chain?
Well, sure, but
What kind of
there's
bond?
there's you know secondary structure, tertiary structure, quaternary structure for proteins.
Right. And quaternary is when it's fully folded.
Quaternary is if you have subunits, but what type of interactions would create like a secondary structure of a protein? So secondary structure is when you have hydrogen bonding between the peptide backbone. Right?
Okay.
So, and then you'll have, so that can produce like alpha helices and beta pleated sheets. So that's secondary structure. What can be there for tertiary?
This one I don't know.
All right. Adding it to the list because this is also very high yield. So
Tertiary, like tertiary is when it's already like a full protein, so it's 3D. So I just don't know what bond. I thought it was the same one.
so it'll be hydrogen bonds between the R groups, also like London dispersion forces, also dipole interactions, but predominantly hydrophobic interactions,
Okay.
meaning if we had a protein that looked like this, and these green regions are hydrophobic, and this is in water, there will be like the water will form a solvation shell around the hydrophobic residues, creating like a rigid kind of cage that has a low entropy because it's very ordered, which means
Mm-hmm.
that there's an entropic penalty here. So the protein will fold in such a way as to sequester or hide the hydrophobic residues on the inside, so they're no longer exposed to the water on the outside.
Yeah.
Okay. Now, there's also a type of covalent bond that can participate in some type of like tertiary or quaternary structure. Um,
Isn't that disulfide?
mhm. And so this question you could have answered by seeing that they're saying subunits are covalently linked. Like subunits indicate that there's a quaternary structure, but also like aside from that, quaternary structure is pretty much the same as tertiary structure. But also, so secondary, tertiary structures have all of those intermolecular bonds, like hydrogen bonds, hydrophobic interactions, but there's one covalent bond that can exist in proteins. Um, and yeah, it'll be disulfide bonds. So for this question, you could have
I
answered
know
it.
that those have to do with cysteine. I just have no idea what thiol means.
Okay, so first we see that they're using the word covalently here.
I know it's either third or fourth.
Oh, so we know that there could only be two covalent bonds in proteins. There's the covalent bond for the primary structures, peptide bond, or there's disulfide bonds. And the peptide bond itself is going to be something that you can't really break um, when you do something like, denaturing or reducing. So this question you could have answered by thinking of disulfide bonds as pretty much the only type of covalent bond in proteins other than the peptide bond. So that could have helped. And also here, when you have gel electrophoresis, right, you can have native, which just means that you're not changing any of the quaternary structure, tertiary structure, or secondary structure. So it makes sense that we have a value of 200 kilodaltons for that. And then in the denaturing condition, it seems like one 200 kilodalton like protein structure is now seems to split into two 100 kilodalton proteins, right?
Yes.
Which is why you see a band at the 100 mark. So that
Mhm.
means that denaturing does mess with some of these bonds, um, but denaturing involves messing with bonds that are those intermolecular bonds, but not disulfide bonds. Because disulfide bonds you can only break by reducing it. So if we now look at the denaturing reducing conditions, we see two bands, like let's say this is like 40 or whatever, and then, oh wait, 60, let's say, for this band, and then 40 for this band. And since we see that if you reduce it, and by the way, reducing like a disulfide bond, let's say squiggle, disulfide bond, squiggle. If you reduce this, you'll get these two, right? And so, okay, we know that this is a hydroxyl.
SH was cysteine, right? Okay.
So we know that an OH group is a hydroxyl group, but it's also an alcohol group. An SH is very similar to that. And that's because sulfur is right below oxygen on the periodic table. So just as this is an alcohol, this was a thiol. The OL ending is similar to the OL ending of alcohols, but the
So it's like the second you know you're working with an SH and you know that it's cysteine, you know that that's identified as like a thiol
yep, so the thigh part is just indicates the presence of sulfur, and the all part OL indicates
alcohol.
the presence of an alcohol, but this is like a combination of the two, so it's a sulfur alcohol, and that can just be called a thiol. Um, so yeah, it can be reduced into the two thiol groups, which means you're breaking the disulfide bond. And if you wanted to reverse it, you can just add an oxidizing agent to recreate that.
Oh, so they're linked by,
What's up?
wait, so they're not linked by a thiol group because you don't have the hydrogen
Wait, sorry. So you can this disulfide bond can be broken by reducing it.
to create a thiol.
Yep, to just you're breaking the
Right,
disulfide
but
bond.
that's not the answer because it's not like automatically as a cysteine. It's a methionine.
No. So let's not look at that right now. So here I'm just showing you that this is our disulfide bond. And when you reduce it, you break the disulfide bond into
Two thiols.
these thiols, right? So another way that we
Uh,
answered this question was to see that denaturing condition compared to reducing conditions. If you reduce it, we get these two bands, which must mean that there was the presence of disulfide bonds, right?
mm-hmm.
Which would mean that these are referring to those disulfide bonds,
Okay.
which would mean which choice?
B.
Mm-hmm. Because cysteine has the thiol group, right?
Yeah.
Methionine has a sulfur attached to like a carbon, right?
Yeah. Okay.
And that wouldn't be a thiol, right? Good. So adding that, to the list. So when
Kyle.
you, oh no, well that, but also like what makes a primary structure, what makes a secondary structure, what makes a tertiary structure. You definitely have to know that stuff.
All right. Cool. Fluorescence.
So what is this question asking us?
What effect does DPC have on the hydrophobic amino acids that are inside of protein X? So they're already hidden. So can't really suppress them because I don't know, D's weird.
Okay.
Hydrolyzes would be breaking a bond by creating H2O, right?
Um, other way, other way
Ligating,
around.
removing H2O.
Oh no. So you're adding.
Maybe adding H2O, so those amino acids wouldn't want that because they're hydrophobic.
Wait, what?
Well, the amino acids are hydrophobic, so you can't add water and it would go up to fluoresce more because it binds to the hydrophobic. Hold on.
Okay.
Hydrophobic surface residues. But hydrophobic like parts of protein want to be inside. Like in a quaternary structure, they want to be like inside and surrounded by hydrophilic. So to bring them to the surface, doesn't it expose them?
Mm-hmm.
So that the more that's exposed, the more it'll fluoresce, and that's why this one's so much higher than either of these, where the hydrophobic are buried inside.
Okay. Wait, go back to that. Sorry. You were saying. Oh, wait. Sorry. Sorry. You were saying that this would make more of them, more of the hydrophobic stuff being on the surface.
Well, yeah, right?
Yeah.
Okay. It's like, is this a trick? Okay, I think I need to read this because, or well, no, this is the first time TDH2 comes up. So cells with a single deletion. Okay. Of the TDH2 gene, which encodes glyceraldehyde 3-phosphate dehydrogenase, or double deletions of the HST3 and TDH2 genes were prepared. The lifespan of each mutant was compared to the lifespan of the HST3 mutant and wild type cells. So we're looking at the triangles.
What type of enzyme class does a dehydrogenase belong to?
Transferase?
Nope. Here, I put something in the chat that, and I'll add this to the list. So yeah, you want to know these enzymes.
Not oxide. Oh, it's an oxidoreductase.
Yep. So you want to know these enzyme classes because like you don't have to specifically know like each enzyme that's doing its thing in whatever glycolysis and
But
Krebs.
haven't we talked about like recently how one of these goes into transferase? Like I remember
Uh-huh.
like it was more than these because we've never spoken about these two.
Yeah, so the most common one would be kinase.
That's what it was, the STY. Okay.
But if it's a dehydrogenase, it's an oxide or reductase, which means that it
Transfers electrons.
yeah, so uh participates in redox essentially.
Okay.
Think about it that way.
Oh, I mean, in the name.
hmm Yeah, but like for instance, dehydrogenase, you would have to know that that's an oxide or reductase, yeah.
Okay. All right.
Mhm.
Okay. So this is the and this is what it's like does r redox to.
Mhm.
Wait, hello. I have no idea what a glycerol looks like, so I'm writing it as the R in the aldehyde. Well, wait, first of all, we know that it's glyceraldehyde 3 phosphate. So even without going through and doing it, we know that it's B.
How do we know it's B?
Oh shoot, that's not the only one it's in. I was just looking at A and I was
But
like,
we
it's
would
not
be
phosphate.
but we would be able to eliminate two choices.
Fructose and phosphoglycerate.
No, so this is in two choices.
Oh, but A and
Get
D
rid of
don't.
A and D.
No, but this one has the glycerol all throughout, and this one goes from like it makes sense. It was remember the one that we were doing it where it was like fructose 6 phosphate to fructose 1,6 bisphosphate. Like it kept the structure and the name. Here it went from fructose to glycerol.
Okay.
So it's I think B if
Mm-hmm.
it's a reversible conversion.
Mm-hmm.
Because that one, if you just take away the phosphate, it would go back to this.
Mm-hmm.
Okay.
One sec. Over Okay. How's this question going?
I went to go like put my laundry into the other one while you were off. We used to analyze the effect of ROS,
Mm-hmm.
reactive oxygen species.
Yeah, they're like free, like free radicals or antioxidants.
Yeah.
So free radicals are also known as reactive oxygen species. It's when you have unpaired electrons, like a single electron on its own, which is super, super unstable, therefore super, super reactive. And antioxidants basically prevent that from forming. So
Okay.
yeah, it says, okay, calorie restriction is known to extend lifespan in yeast. It may exert its effects by reducing the level of ROS as a result of slowed energy metabolism. Researchers investigated the roles of energy metabolism and histone acetylation level, okay, in regulating the lifespan of yeast. What can you tell me about like histone acetylation, I guess?
Histones is a type of protein, right?
Mhm.
Uh, isn't it like the N chromatin?
So, um,
I can't remember acetylation versus deacetylation. That's when it like
well,
comes
tell me a little bit more about the histone.
about the histone itself.
Mhm. Yeah, like what, like, is it like, what is it? Like, of course, it's a protein, but,
It's in the nucleus.
okay, any more?
I don't know.
All right, so I guess you're
I
just,
know it has to do with chromosomes and their structure, but I can't like
Mhm.
get the
So,
DNA then.
so, uh, so I guess they didn't go over it in your genetics class either, but pretty much they are proteins that DNA can wrap around like beads on a string.
Mhm.
And the spaces between the histones have exposed DNA. And if the DNA is exposed, it can be transcribed into RNA.
Okay.
And so if we took that and let's say we methylated it, it would make the histones closer together, which means less exposed DNA, which means less RNA transcription, which means less expression of a gene. Alternatively, you can acetylate it, and that would make the space between the histones longer, meaning more of the DNA is exposed, and that means more of the DNA can be expressed.
If you acetylate them and methylating them is making them closer.
Yep.
Is there another one that's like deacetylation or something?
Yeah, that could just, reverse that.
Okay. All right.
but you really didn't go for this in your genetic class either.
Genetics? No, this is bio, I feel like.
Well, I mean, genetics is a topic in bio, but this is
Yeah.
literally like genetics. Like so far, like I guess they didn't go in your class. They didn't go over anything that's really relevant, but this is a major.
I mean, biochem too. They didn't make us learn like amino acid structures, like names, anything.
That's super weird. Because what else is biochem if not that? Like, what did you learn if not like if a genetics class doesn't teach you what purines and pyrimidines are or histones, like what did you like?
Because it was like all about like mice and like solving the like phenotype and genotypes. Like it was literally just like problem sets.
I know. I'm just saying like it's weird that they didn't teach you any relevant genetics in a class called genetics, and that they didn't teach you any relevant biochem in a class called biochem. But because like biochem like is
Because it's like I take
fundamentally.
courses that are like, for example, biochem here is a 2 semester course, and I took the condensed one that's like 1 semester.
Mhm.
Like, so it goes through it super fast paced, and you do a lot of
But like if you take
like
any
they don't want us to be spending a time like with the calculation and memorization and more conceptually understanding the topics. And the MCAT is very like you
well,
need to
so
know
you said
this and
it's conceptual, and there's no way you can teach biochem or genetics like regardless of whether it's conceptual or not, like without teaching you about histones and pure like if they don't like if so
I
DNA
can
is
show
made
you. Like,
our DNA is made up of purines and pyrimidines. So I think that if a genetics class doesn't teach you that, it doesn't really teach you genetics. And same thing with biochem. If you take any biochem textbook, like they'll talk to you right away about amino acids and
well,
things.
we talked about amino acids, but it was like,
Oh, you said that they didn't teach you anything about amino acids.
no, we just didn't have to memorize them.
Oh,
Like,
but I mean,
it's like, why does DNA exist? Chemical composition. We learned the functional groups.
hmm?
Um,
It's I'm not saying to memorize them. I'm saying like if you don't know.
like this is what I learned about.
Yeah, so I'm saying if you don't know anything about how amino acids can form a protein, like
No, that I knew. I knew like primary, secondary, tertiary, quaternary structure. I just didn't know like
we just went over it though, but you didn't know.
because we never went specific into what kind of bond goes with each one. That was never ever
What did
discussed.
they teach if they didn't teach that? Like if they like look, like if a DNA, if a genetics class doesn't teach you what a pyrimidine or a purine, looks like.
This is like the one time we went over dimers was this.
Uh-huh.
This sentence, that was the entire thing with dimers for this
Yeah, but,
semester.
but I'm saying if they're talking about thymine, but they don't teach you that thymine, you know, is a pyrimidine and that thymine has a single ring, right? That's kind of like not a useful class. And also if they don't teach you in biochem like what a protein, because that's if you take I guarantee you take any biochem textbook, first chapter will talk about amino acids. First chapter will talk about proteins. And you know,
Mm.
that's pretty important. So kind of sucks for you, but yeah, you definitely need to know that stuff. So I don't know what they're trying to do here.
I don't know. So yeah, that was like amino acids.
Hmm.
Wait, thought I just saw it. Never mind. Like all of this stuff I know. I just like it's really not as in depth as whatever.
Yeah, I mean it
Why
seems
it doesn't carry over to MCAT well.
wait, no, I mean, well, I guess just like I guess the classes that you took. just because if you take any, like, if you just take, maybe, like, you, even if you look at the biochem textbook that you used, I guarantee you in, like, in the first chapter, they'll talk about protein secondary structure, protein folding, because that's pretty much like,
We don't use our textbooks that we go off of lectures.
yeah, that's probably why, probably why. But yeah, that sucks because it's not like it's. But you would imagine biochem, a class in biochem or a class in genetics to teach you the very, very basic stuff, but it sucks that they didn't, because now you're just kind of learning the stuff for the first time, and that's not good.
Yeah. Um, so wouldn't it be these two, the one that has the antioxidant versus doesn't have it, so that we can like see what having them
Mhm.
will do? But these are both negative, so we would only want to look at like what does hydrogen peroxide have to do with it? OH?
Um,
Mm.
so hydrogen peroxide can undergo a free radical like decomposition, something like this, where a single electron will go there, a single electron will go here, and you can have your free radical OH group. So that's not an OH minus, it's just an OH that has that's a free radical because it has an unpaired electron. So that can be something that if you have some reactive oxygen species, let's call it X, it with a single electron, like as a free radical, right? The This can form a bond with it, making it like XOH, but important thing that's happening here is just that we're getting now like a lone pair or a pair of electrons as opposed to an electron on its own. So that could the hydrogen peroxide could help reduce it could be in like an antioxidant for the ROS because it can decompose in a way that would produce free radicals.
So we would want to see how like the presence or absence of hydrogen peroxide would affect the free radicals.
Mhm.
So they're all three of them are possible solutions.
Mhm.
Okay, so let me explain this back to you.
Mhm.
ROS are the free radicals that can be counteracted with antioxidants, and that's why we'd want to look at the wild types because it would test out like how the ROS would affect the yeast in its absence and present, and overexpressed what that would do too. If too much will also ruin it.
Mhm.
And then the hydrogen peroxide essentially like attacks the free radical.
Mhm.
So isn't that similar to the antioxidant structurally?
Yep, it can
Okay.
yep.
All right. So cool. Vasopressin regulates the insertion of aquaporins into the apical membranes of the epithelial. So I'm just gonna tell you which of these words I don't know. I remember we went over this when we did the
Mhm.
like when we did veins, like when we were explaining capillary artery. I feel like you were saying something about this
Oh,
passage that was in.
so this is about the nephron. that we talked about, but
aquaporins.
like, what do you remember from about the nephron? That's another good thing to know.
Um, that was part of the kidney.
Mm-hmm.
That was when we were doing like the which part would be
Yep. Do
um
you think?
which part would be the collecting duct and like where would the urine, the most concentrated urine go?
Mm-hmm. So can you let's see if you could draw like a nephron structure.
I feel like it was just squiggly lines and it went down and then it was more squiggly lines. And then there was like a part that was in the like
So yeah, try it out.
okay, so we had this and then these.
Mm-hmm.
There's something else.
Mm-hmm. Okay.
Like
So where do you think the stuff in the choices are on your drawing?
let's say this is the collecting duct.
Good.
This is proximal and distal
Yep.
tubules.
Mm-hmm.
Ascending would mean that it's going up, so this.
And then the descending on the left. Good job. So
Men's capsule isn't part of this, though.
it is here. So this is your Bowman's capsule, and this is your glomerulus, which is just a bunch of these arterioles and stuff like blood vessels. Because what's happening here at the Bowman's capsule is the blood is being filtered through from the glomerulus into the Bowman's capsule. So that's the first thing, right? And yeah, and then you have your proximal convoluted tubule here, your descending loop of Henle, ascending, distal convoluted tubule, and then yeah, your collecting duct here.
So that's the last step.
And now, do you remember anything about like this dotted line here?
Um, it was medulla and cortex.
Yep. So cortex outer portion, medulla inner portion, and there's one other thing that's special here that I guess highlights the difference between the cortex and the medulla.
Uh, um,
So as you go from the cortex to the medulla, and as you go deeper and deeper, you're going to be in a more concentrated solute environment,
right.
which means that you're going to be in a environment with a higher like osmolarity. So that's why, like for instance, in the descending loop of Henle, you have water getting sucked out like through diffusion, like without needing ATP. So like that's also kind of the thing, like when we talked about the sodium-potassium pump for neurons that uses ATP, like if something is moving against the gradient, like well, if something is moving with the gradient, aka from high concentration to low concentration, that's just normal. That just happens without any, you don't need to add energy to that because it just happens naturally, right?
Okay.
That'll just be diffusion. So if something goes along its concentration gradient, it's going to be diffusion and it won't require energy input. But if something's moving against its concentration gradient,
Active transport.
yep. So yeah, this H2O is just diffusing out because as we go deeper into the medulla, the solute concentration is higher outside, which means the H2O concentration is lower outside, so it sucks the H2O
Oh,
out.
right. So it's super concentrated.
So yeah, and also if it's coming out, there's the vasa recta, which is just the capillary network all around this nephron that we typically don't draw just because it's like going to be messy to draw. But you should know that when this water comes out, or if anything comes out, it's to go back into the bloodstream. So that's why we call it reabsorption. If it comes out of the nephron tubules, it's reabsorption, meaning reabsorbed into our bloodstream. Whereas if it's going in, it's called filtration, which means it's not going, it's leaving our bloodstream and it's going into this filtrate, which ultimately leaves the body as urine.
Okay.
So good. So good
I feel
job.
like I remembered a lot of this.
What's up?
You have to give me credit. I remembered a lot of this.
Yeah, I was just gonna try say that yeah, you got this. So I think it's a good strategy for you.
I still don't understand what aquaporins, that's like when the H2O is still there because
So
water pores.
before that, I just want to say that so it seems like it's going to be a good strategy for you to, if you don't remember something, just start drawing it. Because when you started to draw this, it was starting to jog your memory, and you were able to tell that, this is the collecting duct, this is the loop of Henle, this is the proximal, this is the distal. So when you don't remember something like that, start drawing it. Same thing with the depolarization, whatever stuff. Well, that's wrong, but you know, that kind of thing. Like, just start drawing it, and I think that'll help you remember it. But yeah, so so vasopressin has another name. Um, its other name is ADH, which stands for antidiuretic hormone. Right?
Okay.
But you could also think of this as aquaporins drilling holes.
What are aquaporins?
What do you think? Let's think about
Water pores?
yeah. So aqua, water, pour ins, pores, like holes.
Right.
So so
So it's water. So I think it has something to do with this.
so it's going to actually work on the collecting duct here, causing causing water to be reabsorbed at the collecting duct. Um, and you can also kind of think about how alcohol or caffeine, they inhibit the release of ADH, which means that since the ADH is causing water to be reabsorbed, if it stops that from happening, we're not going to be reabsorbing the water, which means it stays in the tubules, which means it leaves as urine. And that's why, like, if you're like drinking alcohol, you're going to dehydrate yourself. You're going to be losing a lot of water. And you need it for the Krebs cycle, and our brains can only really use glucose. It could use ketone bodies as well, but that's not something that is easy to do. So that's why like the hangover feeling with like the headache and stuff is something that happens. It's because you're essentially dehydrating yourself, yeah?
Cool. So it's collecting duct.
Yes.
Okay, but cool that I remembered everything but Bowman's.
Mm-hmm. Look at all these math questions in the BB section. What are they doing here?
Okay, let's
Mhm.
see what values they gave us just to plug in. So we have I think. Why is it? Okay, so I want to say P because that's a Russian P, but I don't know what it is. Let's say 24. We know temperature is 25 Celsius. I is 2
Does it make sense why I is 2?
because 2, wait, don't 3 sodiums leave?
No, you're thinking of sodium-potassium pump, which is different. But this is saying what has an I of 2?
I don't know what Van 't Hoff is.
So, yeah, I'm asking you like, so they're giving it to us, giving us the value, the Van Hoff factor for what?
For sodium chloride.
Okay. So, I mean, we did go over this before, but let's see if you can figure it out from us being given this information.
Oh, is it like distance from the ideal gas?
Uh,
Like,
no.
no, doesn't have to do with like electrons and like charge. Um, it's not like a number, a numeric value, but that like Na is 2 plus Cl is minus. So it'd be +1, not 2. I have
Uh,
no idea. Just because there's 2 of the
uh-huh.
like number of molecules.
Uh-huh.
That's it? Just
So this turns into what?
Na plus Cl.
Cl minus, right?
Right.
So it's like, so if we think about this equation, right? The M is the concentration of the solute, right? Right.
Yeah.
So the solute, let's say if the solute is NaCl and whatever, let's say it's like 5 molar. When it dissociates in water, it's going to be, each of these will be 5 molar, right?
Yeah.
So we're going from 5 molar to a total of 10 molar when we dissociate the sodium chloride, right?
Yeah. So it's
So,
double.
yep. So the Van Hoff factor is to account for that. Because if we didn't have it, what would we plug in for molarity, right? We'd be plugging in like the 5 value, right?
Mm-hmm.
So
Cool. I did not remember this at all though.
I know. But what would it be if it was this?
3.
Yep. Because it breaks apart into 3 different things, right?
Mm-hmm.
And so, if they told us this was 2 molar, like the entire point of this, right, is that if they tell us that this is 2 molar, we can plug in for capital M 2 molar, right? But the fact that this dissociates into 3 things means that we're going to end up with more than that.
Okay.
So what did I just say?
You said that because we end up with three things, we're going to get more than 2 molar. So we have to account
Mm-hmm.
for the difference with the I value.
Mm-hmm. If
That's a Van Hoff factor.
what would the Van Hoff factor be for glucose?
24.
24.
C6, H12O6.
So
6 plus 6 is 12.
glucose breaks apart into 24 things.
Well, not if you count for like OH and like
So
there's a row that's like CH3 and so it's less.
mm,
But do you like actually have to sit there and
no. So when you put glucose in water, what happens? Because you're saying it breaks apart into 24 things. But is that what happens when you put glucose in water? Does it break apart into 24 different things?
no?
Okay. So,
I can't like think of any reaction that happens when glucose is in water.
mm,
Don't we just write it then as aqueous?
yeah,
So
but
nothing happens. So it's just there is no I value.
it'll just be I is 1, right?
But why is that? Oh, I thought it would be I is 0 because it doesn't change, but I guess yeah, 1.
I is 0, then this value is 0.
Okay.
But that's not going to be a value that's possible, right?
Yeah.
So, so these ionic salts, when you put them in water, they dissociate. Glucose is not an ionic salt, so when you put it in water, it's not going to dissociate. It'll just remain glucose.
Okay.
But you want to,
Make sure I know this. I know.
yeah, because when you said 24, like, I don't know, like, because that would mean glucose would break apart into 24 different things, right? Okay. All right. So yeah, let's try this out.
So I is 2. Oh, shoot. I lost my annotate. So, and we're looking for M.
Yeah.
R is, oh, I forgot to write that out. I can tell from R if there's something weird with the ATM is here.
Mhm.
Is it temperature? Do I have to convert it?
What do you think?
I just want to solve it first.
Okay.
So you isolate M, we get RTI over 24.
Yeah.
40 times 0.8 is 0.3
Yeah.
over 24.
It might help to draw the units for each of these values.
No, that's what I was like. This one's ATM. It's just so hard to draw on this stupid thing. See.
I mean, if you want,
Oh, this is a 25, not a 20. So this is 50 times 0.8. It's 0.4.
so you can.
It's still way too small. I don't know what the unit for I is.
Well,
There is one. This is the long one. L times ATM
mhm,
over mole times K.
mhm,
Isn't M the same as moles over liters?
uh-huh. But from what you just wrote, it can
Liters per
help us.
mole. So we can flip this. Or
So
wait, no, well, this and this, we divided by R. So that means this and this cancel out.
yeah,
K is Kelvin.
mhm,
And the conversion rate, it's like 340 Kelvin, 375. I can't remember. I know that value 375 Kelvin's important. I just can't remember. It was that like
so
boil
so can we? So we could at least agree that we want
say
to put
yes, we need to change 25 Celsius.
good, good, good. So yeah, so it's 273 plus the 25 Celsius. But what can help you remember is that absolute zero, or rather, if something's in Kelvin, well, temperatures in Kelvin can't be negative. So I guess if you think about some value in, well, I'll just say to just remember it then. So Kelvin is 273 plus whatever the temperature is in Celsius. I was going to say that 0 Kelvin is absolute zero. Can't get lower than that, but
Right.
but that may not help in remembering this. But yeah, just kind of remember that. Maybe it could help to remember that 0 Celsius is 273 Kelvin, but yeah, at least you're able to tell that we want to convert.
375. I think flipped it basically, but whatever.
Yeah, but yeah, at least you're able to tell that we need to change the 25 Celsius into something in Kelvin. Yeah. So this will be 298 Kelvin, which is, you could just say 300 Kelvin.
Okay.
Mhm.
So
So we
300 times 2, we have 600.
24 ATM over 2 times 0.08 times the 300 Kelvin.
Isn't this 0.2, basically 0.16?
Sorry, what are you saying?
2 times 0.08 is 0.16
Yeah.
times 300. Would that be like 48? Because it would go 2 decimal points back.
Uh,
3 times 16 is 48.
so you're saying,
I think it's 24 over 48, and then it's 1/2, unless they like lost a decimal point,
yep, you're right.
which is B. Okay, so I was kind of excited that they gave me all the values, and I knew it. Like I looked at R and I was like, there's a trick. But I like just went ahead and did it anyways and saw that it didn't give me the right answer and knew that I had to go back and figure out where I went wrong. And that was with Kelvin and Celsius.
Yep, you always want to make sure that all of the units are accounted for, because otherwise, if you did the if this was 25 Celsius,
It
you would,
wasn't working. It gave me like the tiniest like 0.
yeah, because the ATM can get canceled there. And if we're looking for our answer as moles per liter, the moles and liter part will also be canceled. So the only thing left is this value in Kelvin, which we're multiplying by a value in Celsius, which is not going to get rid of that unit. So that means we have to use Kelvin here in order for the kelvins to be canceled out.
Okay. Yeah, I got it. Okay.
Just
I feel like I can't remember how many I got wrong in this. No, okay. So none. Small intestine, large kidney. So if it enters through the small intestine, the next one is large intestine.
uh what are they saying here in the question?
If it entered in the small and enters the blood stream.
Yeah. Always remember to read the question.
So where does the from the small intestine go?
So it says it goes from the small intestine into what?
The bloodstream.
Yep. So it can't be large intestine because
But isn't like, but if it enters, we'd want it to be like deoxygenated blood, right? So
uh
at what point does like I'm thinking if it enters a vein or not. Like it would because the blood's deoxygenated. Because
uh
it's already, like been through the body.
so let's think about which organ.
It's like a waste.
So if we have our small intestine and then we have some blood, ultimately what organ could this be, right? Now we know that it can't be the large intestine because that's where it would go if it didn't go to the blood, if it just went from, the inside of the small intestine into the inside of the large intestine. So we could at least get rid of that. And if it went this way, it would be going into what?
Stomach.
So it can't be that either because they're telling us that it has to go through the blood first.
Mm-hmm.
Now, Did anything really enter into the pancreas?
No.
No, right? The pancreas can make hormones that can enter and affect other things, but
If this was a short response, though, I feel like I would have picked kidney. Because I feel like all the bad stuff goes there to be filtered.
well, that would be wrong.
Yeah.
So,
But I don't understand, like, how do we, just from like crossing it out, like, which would be the first major organ?
well, yeah, so I'm showing you like a way that
That's
to
right.
answer this, like if you didn't know, like where it actually goes. So we know that large intestine is if it continued to go without the blood as that middle part, and stomach would be if it continued to go backwards without going through the blood. So that's why we can eliminate A and C. And then pancreas, we know that that could make hormones that are important, but nothing really goes through the pancreas, right?
Mm-hmm.
So the only one that's left is the liver. So that's how you would answer this question if you didn't know the answer, right? But also now I can tell you that the blood that so when we have, stuff from our small intestine uh getting taken up by the bloodstream, they will go through this hepatic portal vein. And a portal vein is essentially a vein, or,
It's right about it being a vein.
what's up?
It's right about it being a vein.
Oh yeah, yep. But it's a portal vein, and a portal vein means that it doesn't go through the regular circulation. Regular circulation would be the stuff going to our heart and then the rest of our bodies. But the portal vein is like a portal that takes it directly into the liver rather than going through the whole body circulation. And you want to do that because bad things can come in, and the liver is where detoxification happens. So it's like if you're absorbing this bacteria or whatever it is, yeah, it's a bacteria that can, they're telling us, cross the epithelial cells of the small intestine. Like it's going into our blood, and but we want to make sure that it's nothing bad that's going into our blood. So we have it go through the liver to detox, and then it can go back into the bloodstream and go through, like, the rest of our body.
Okay, that's cool. So the portal vein takes it out.
Yep, hepatic portal vein.
Hepatic. Okay.
Yeah. But also, if you didn't know that, that's how the logic
Okay,
part can
so
go.
nothing goes into the pancreas, stomach, and large intestine is where it would go normally.
Yep.
Isn't it then kidney, pancreas, liver? Or pancreas, kidney, liver?
For what?
Like that's the last step. Like where does it go after the large intestine?
You're saying if it's not taken up by the blood,
Yeah.
it will just exit out of your colon. Um,
All right.
and,
Okay.
and but also certain things would get absorbed in your large intestine, like water and vitamins and nutrients and stuff like that.
That we can absorb, which are base. Like we can absorb B, C, D, and K.
Um,
Or like salt like that we can absorb.
uh,
I have it in a book. Hold on.
okay. But yeah, that won't be really something that,
Okay, not important.
like, I guess they'll either be the ones that are fat soluble or the ones that are water soluble. So if it enters into the blood, it would be the water soluble ones, but if it enters into through the lymphatic system, it'll be the fat soluble ones.
Okay. All right, forget it.
Mhm.
I'm not gonna find it quickly. Okay, never mind. So it's just based off of this.
Mhm.
From I'm like
Mhm.
oh wait, it's this one. And so I said it's almost like 4 to 8. But I should probably just calculate it. Right? Because I'm like 88% sure I picked D
Mhm.
last time.
And
It was wrong, obviously. It's on this
get
list.
that.
Huh?
Oh, like, so how do you get that?
Well, I just approximated based off of these two values because amplified pituitary responses to GNY. So we're looking at just GnRH and then we're looking at it plus the NPY.
Yeah, but I'm asking how do you get this value?
I guess that's like really wrong now that I look about at it. It would be less than 50%. And since it's even smaller, that was a really stupid mistake. It's like 5/7.
Okay. So, yep. Just remember, do the math. Don't want to not do the math. So just got to do the math.
I know.
So, yep. What would your answer be?
A. It's like 5/7.
You could try doing the math.
I am. 5 times 100 is 500 divided by 7. It's going to be something like 70%. 100 minus 70 is A.
Not sure what you did exactly with the math. Like, what are you doing?
Instead of 4.74, I took 5.
Okay.
And here I did 7. It's 5/7 percentage. 5 times 100 divided by 7.
Why don't you do this?
I don't know. That's just, but I got the right answer, no?
No. Like
Okay,
they're
so
asking how any if
what
you
does 7/5 give us? I don't understand. You do 7
look, they're asking us so they're asking us if we just had GnRH
be 5.
and if we add NPY, by what percent does this increase?
Right.
Mhm.
So you want to find what percentage of this total is this, so you can subtract it. If you do 7/5, 7 times 100 is 700 divided by 5. That's going to be way over 100. That's like 140.
Okay.
So it's 40%.
So if it's so you're saying like it's 140% increase, or it's 140% of
The total.
this is 100%, this is 140%, right?
Yeah,
So it increases by
40%.
amount. And since I made I rounded up for the denominator, our answer here is smaller than it should be. So our answer should be a little bit higher than 40%. So you got to do the math. Because you got the wrong answer and then you did it again. You got another wrong answer. So
I literally did do the math.
mm,
This time I was sitting there and doing it. I did 4/7, 5/7.
Yeah, but if you're so they're asking us by what percent does this increase by, right? So if you're doing 5/7, right?
That's the amount. But wouldn't it be like, wouldn't you subtract? Like, it would give you 71%.
Well, you want to see by what percent does this go to the 7, the 4.74?
Okay.
By what percent will it increase to get to the 7.03?
All right. Fine. B.
Mm-hmm.
Should we read it?
Mm-hmm.
Ebola virus causes a rapidly progressing and often fatal hemorrhagic disease that currently has no effective treatment. How long ago was this written? Ebola virus particles consist of nucleocapsid encased in a membrane that contains transmembrane viral glycoprotein EGP. The virus has a single-stranded negative sense RNA genome that is used as a template for the mRNAs produced by a virally encoded RNA-dependent RNA polymerase. Single-stranded. What does negative sense mean? Like that it's
I think it's like the non-coding strand. Let me see.
so it's 3' to 5'. Because it codes 5 to 3, or it's read 3. Okay, no, so it's 5 to 3.
Oh, it's the template strand. So it's like the thing that's complementary to the actual RNA. I believe so. Let me make sure. Positive sense RNA virus, type of single-stranded RNA viruses whose genetic material is viral mRNA that encodes for proteins. Negative sense RNA virus, single-stranded RNA viruses whose genetic material is the antisense strand of the viral mRNA, and that's just the one that's complementary. So the coding strand itself would be the positive sense, and the negative sense would be the template strand.
Okay,
Mm-hmm.
all right. Use that, right, because it's used as a template for the mRNAs by a virally encoded RNA-dependent polymerase. The entry of the nucleocapsid into the host cell's cytoplasm starts with the fusion of viral membrane with a host membrane, process facilitated by viral glycoprotein. Recognizing the similarities between how EGP and the glycoproteins of related viruses are processed during viral infection, as well as how inhibitors of endosomal acidification block EBOV infection, researchers hypothesized that acid-dependent proteases trigger the conformational changes in EGP that are necessary for fusion. The infectivity of VSV EGP vesicular stomatitis virus particles engineered to contain EGP instead of VSV glycoprotein in the viral envelope was reduced more than 99-fold by inhibitors of the mammalian proteases CAT-B and CAT-L. In separate experiment using a strain of Ebola, CAT-B inhibitors reduced the number of Ebola particles produced in a cell structure 10-fold. Figure 1 shows how the introduction of certain genes into 2 mutant mouse cell lines affected the subsequent infection of those cells by VSV EGP.
Okay, let's take a look at this figure. We have y-axis total or percent of total cells infected, and x-axis introduced genes. So when you introduce no gene, aka wild type, you get this 27, 26% value. And if you have the bacterial gene, it's like nothing. Oh, this is with the some version of it that doesn't have cat B, but has cat L. And if you add cat B to it, you get a really high percent of infected cells, right?
But are these the cat B, cat L themselves or the inhibitors?
Um, Inhibitors.
You see, it says
Mhm,
9 reduced by the
mhm. Yeah, I think
that's
these
that's
are just talking about the cat B proteases. Because otherwise it seems like they're saying that the infectivity when they used inhibitors got reduced. So cat B inhibitors reduce the number of the Ebola virus particles produced in the cell culture tenfold. So cat B, if it's inhibited, it reduces the viruses, but if it's not inhibited, if it's actually there, it seems like it makes more of the virus or results in more infectivity,
okay.
right? Just cat B inhibitors reduce the number of the Ebola virus particles 10 times. So if you inhibit cat B, you'd have less of the virus, right?
Yeah.
Yep. And so here we see that we knocked out cat B, but we keep cat L, and in this bacterial whatever, it's like really low. So pretty much not causing any infection. But when you introduce the gene for cat B, you thereby make it so that it makes cat B. We have a much higher percentage of cells that are infected, right? So yeah, we can say cat B causes more of this virus, infectivity, right? And then if we just have cat L, which we already have, but no cat B, it goes back to like nothing, right?
So
In
it's
DY.
related to infectivity.
Mhm. And then in the when we knock out the cat L and cat B, as opposed to knocking out just cat B, we
Oh, I didn't see that. So the pluses.
mhm,
Okay, all right, I understand.
yep. So yeah, so okay, if we don't have cat L or cat B, we get low amounts, but if we introduce cat B, it goes up again. And if we introduce both cat B and cat L, it goes up even more. So so yeah, cat B proportional to virus infectivity. Um, but it does that even more so if you have cat B and cat L,
Okay.
even more.
Hmm. All right, that makes sense.
Mhm. Yep. That's probably all we need for this question.
Okay. Is cat L? No,
Yeah.
because cat L on its own was low.
Hmm.
Is C true? Does not infect cat B cells expressing cat L. There's no that's like,
So let's see. C says no because VSV-EGP does not infect cat B like negative, like cells that don't produce cat B. It does not infect the cat B knockout cells expressing cat L better than it infects the cat B knockout cells not expressing cat L. So cat B minus that expresses cat L is this, right?
yeah,
That's what, so that's cat B minus expressing cat L compared to cat B minus not expressing cat L. So I guess we're comparing these two.
right. So cat B expressing cat L.
Seems to be causing more infection.
Action. So
The cat B minus not expressing cat L.
A, right?
That would be it then.
Or because it's not enough cat L on its own. So it has to be a no. And we know that it does do this. So it has to be D.
I think that it
X cells expressing both cat B and cat L. But I thought that this means that cat B isn't oh, hold on. It's not expressing extra cat B because cat B is already present, so it's only doing cat L, and it's skyrocketing. It's expressing both cat B and cat L better than cells expressing only cat B without cat L. So isn't it D?
well, so what were you saying about this?
That makes it more infectious.
So that's when that's when it's cat B minus that expresses cat B through that introduced gene.
Yes.
So this is saying that cat B plus, so cells expressing both cat B and cat L. I believe that would be the
A.
wild type, I believe.
Oh. But C's wrong. Not infect cat B cells expressing only cat L better does not infect. It does infect it better.
So the cat B minus cell that expresses cat L is this, but it's not this bar because this bar has cat B as the introduced gene. So that's what this bar represents.
I'm confused.
So
It says cat B cell,
B minus, right?
right?
Minus means it's not there, right?
So it would be like this or
Yeah, it'll be like this or this, but it won't be this one because this is if you introduce the cat B cell. If you introduce the cat B, which we don't have in this knockout,
okay.
right?
This is
So
getting more confusing.
Yeah. So like I think what will help if we think about like okay, so cat B minus cells that express cat L is this. This shows cat B is not there, but cat L is there.
Mhm.
So we're looking at these compared to when they say B minus cells that are not expressing cat L, which will be this, right?
Right.
So this is when you don't have cat B and you don't have cat L. Whereas this
Okay.
is if you don't have cat B but you have cat L. And it seems like when you don't have cat B but you do have cat L is not producing a higher infection compared to if you don't have cat B and you don't have cat L. Because if you don't have cat B or cat L, it seems like you're having a higher amount of infectivity.
Okay.
So yeah, it's tough because it's a lot of stuff being thrown at you. Uh, but I think you did well by initially deciding that it's not sufficient. So we can get rid of A and B. Um, but yeah.
So reduced.
Wait, why?
EGP was reduced 99-fold by cat B and cat L.
You mean if you inhibited cat B?
Yeah.
So that's if you inhibit cat B, but they're asking if you don't inhibit it, what does cat B and cat L do?
So it would be the opposite. It would
So what does it say that cat B and cat L like do?
they're proteases.
So what do proteases do?
See?
Hmm? So what do the proteases do?
Like breaks peptide bonds down into smaller ones.
Yep, it breaks apart proteins, right?
Yeah. I can't remember like
Mhm.
okay, well, ooh. I like the keypad. I like brush my finger against it and it goes insane.
$44.
Why isn't it?
Uh-huh. Yeah.
Okay.
So,
I know that the two like sugars are there.
um, so,
Like succinate comes, there's like some sus acid,
so
like the step before it.
let's think about glycolysis first. What's the starting material in glycolysis and what's the end material?
Glucose,
Mm-hmm.
and you end with pyruvate. So you wouldn't want this to be the ending here.
Mm-hmm.
So D's wrong.
Yep. Which means D is right?
Yeah.
Yep.
So this one is like here in the circle,
Yeah. Mm-hmm.
and then succinate's here.
Mm-hmm.
And I don't remember where malate is, but it's somewhere on the bottom too.
Yeah, I think it's something like this.
And
Yep.
K is here.
So yeah, that could be a good way to. So if you didn't remember, all of this stuff for the citric acid.
I knew that these two weren't in there. I wasn't sure about C.
Yep. So that's why.
I think that's what I picked last time.
Yep. So that's why I asked you about uh
Glucose.
first, because then it helped jog your memory of that it's breaking down glucose into pyruvate, and if if that's something that's involving pyruvate, then we know that that'll be involved in glycolysis and therefore not involved in TCA. Yep.
Okay. Oh, okay. Do you know I hate the reading ones? I wish they were all discrete.
Hmm. Yep.
Cell types including cytotoxic T lymphocytes and skin melanocytes use modified lysosomes called secretory lysosomes to secrete cell-specific products in response to appropriate stimuli. Consequently, in mice and humans, mutations that disrupt cellular machinery specific to the secretory pathway can cause immunodeficiency and partial albinism. CTLs use secretory lysosomes called lytic granules to secrete cytolytic proteins. Melanocytes use secretory lysosomes called melanosomes to transport melanin pigments. Melanosomes are transported along microtubules to the cell periphery, where the melanosomes transfer microfilaments prior to the release of melanin from the wall. RAB27A proteins on the melanosome membrane facilitates this transfer by interacting with melanophyllin protein, which in turn binds myosin VA, a motor protein, on microfilaments. It's like so much is pushed into you in like one sentence. People with Griscelli syndrome have hypopigmented skin and silver gray hair. These pigmentation phenotypes are sometimes but not always associated with a immunodeficiency resulting from a defect in the killing function of CTL. Characteristics of two mutant mouse lines that are used as models for Griscelli syndrome are shown in Table 1.
All right, we see mouse mutation ashen causes RAB27A to be inactivated, and there seems to be no more killing by STLs, the cytotoxic T cells, but there does seem to be a partial albinism. Okay.
Mm-hmm.
Dilute, if the mouse mutation is dilute, it inactivates myosin VA, which lets stuff get killed by the CTLs as well as produce partial albinism. And okay. Given that secretory lysosomes form normally in the melanocytes and CTLs of ashen and dilute mice, the data in Table 1 best support the conclusion that melanosome secretion and lytic granule secretion differ in that the secretion of lytic granules does not require. All right.
So let's see where lytic granules. Melanosome had to do with Rab27. And
So CTLs.
I see that I forgot I can't find the line where it said. Lytic granules. It was somewhere. I can't find it.
What are you looking for?
But if CTL is positively associated with lytic granules and they're absent, then Rab27A is not required.
Let's see. If you mess up Rab27A, you don't get killing by CTLs. And it seems like CTLs from here use lytic granules, right? So Rab27A, if that gets inactivated, it's not, the CTLs aren't able to kill stuff, right?
Right.
And CTLs need
Good thing for them to be killing stuff?
What's up?
Is it a good thing for them to be killing stuff?
I guess no, because I guess they're trying to kill some bad stuff. I don't know if it's good or not, but I know that it seems that CTLs use lytic granules to kill stuff. And if there's an inactivated Rab27A, there's not any killing by CTLs, which should mean that we're not getting lytic granules, right? But if we have an inactivated myosin VA, we have killing by CTLs take place, which must mean that lytic granules still get produced if you inactivate myosin VA. So it doesn't matter if this gets inactivated or not. You'll still get killing by CTLs, whereas the Rab27A, if you don't have it, like if it's inactivated,
So we need the Rab20, but we don't need the myosin VA. So it's the inverse of the table. Oh, Shane, I can't anymore.
We're pretty much done anyway. So
just go through to the end, however many there are left. Oh, wow. Okay. Transcription factors FOXP3 is encoded by an X-linked gene in mice and humans. Approximately 60% of female FOXP3, whatever. which carry one inactivating scurfin allele and one wild type allele of the FOXP3 gene, spontaneously develop mammary tumors by 2 years of age. Following experimental observations clarify the relationship between FOXP3 development and the development of mammary tumors. Easily detected in non-cancerous female mice. Undetectable.
I think we can answer this without that.
Okay. Those tumors overexpress okay. Well, no, because this is the first time that this promoter came up.
So we're being told in this question that you can make an antibody that's developed to be highly specific for the extracellular domain of HER2.
Okay.
And it's just asking where is that domain located.
I have no idea.
So we have an antibody here. Where on the antibody would it bind to like antigens or something? All the way down there?
No? It feels like I don't understand what the structure is. Are these like entrances?
Oh, like an antibody? Right?
Okay. So like here.
Mm-hmm.
So would it bind to the black and the stripes?
Mm-hmm.
So 1 and 3.
Yep. Because those are the variable like so in an antibody they're like Y-shaped, right? And the they have variable domains here, which is from alternative splicing, but it also means that because we use antibodies when we have a specific antigen that we're trying to destroy, and it's essentially just asking us which region is going to be
Right.
specific to the
And
specific thing that we're trying to destroy, and it'll be the variable region. So, yep. So, yeah.
what was the like funny saying to remember
7 up.
7 up? But that's just to remember the parts of it.
Mm-hmm.
Is it like in the
Oh, I see what you're saying. Got it. Yeah, so the urethra is just the opening. So, and the urethra is the opening where like pee also comes out, so it shouldn't be a special region where gametes become.
Well, yeah, I'm like
Uh,
pretty sure I said A.
mhm. Now, the prostate gland is going to release like the semen, but not the sperm. So that should be out. And then the testes is where the, I guess, sperm,
Sperm is stored.
immature sperm is made. But yeah, it'll be they'll
Epidermis.
they'll mature in the epididymis, being motile, which means they can move and capable of fertilization. So, yep, it'll just be that.
Interesting. Okay.
Yeah. Oh yeah, I think we talked about this, or yeah, the reason for rigor mortis, right?
Yeah, but
Mhm. So we know that in the cross-bridge cycle that we have our actin and our myosin.
I know that ATP can't be produced, so I think that A and B are wrong. And
Okay, yep.
wait, but continue with what you're saying, because I don't know.
So yeah, the myosin head like binds to the actin when the ADP, sorry, when the ATP can like hydrolyze to form like the ADP and the phosphate. So that's what causes it to bind to the actin, and then you can do the cross-bridge cycle and stuff. But you need ATP to, well, if ATP
Do
being,
this
what's up?
to make the cross bridge.
So if the ATP gets used, like hydrolyzed, it will make the myosin head bind to the actin filament. But you need ATP to detach it, to detach the myosin head from the actin filament. So that's why when animals die, they get rigor mortis because their myosin in their muscle is still bound to the actin. So it's stuck in that contracted state. So
Okay.
basically ATP breaking into ADP plus inorganic phosphate causes the attachment of the myosin head to the actin, whereas the opposite, or having ATP would cause it to detach. So the ATP helps the actin, or rather the myosin head, to detach from the actin filament.
Okay.
And just remember, because it's not the most intuitive, right? So just kind of remember that connection with rigor mortis. Like if you're dead, you're not going to be
And
bring.
separate the myosin from the actin. But
Yeah, if you're
that the
dead.
myosin is as the head.
Mhm. Yeah.
So
So if you're dead, you can't bring in new ATP, and you need ATP to detach the myosin head from the actin filament. Yeah. Right.
this one was confusing because
So this is kind of like the first problem we did today. Um, so they're showing, so we have a homodimer, which means it's made of two things, like two monomers that are the same. And when they use SDS page, which is just denaturing, they get some value that's at 45 kilodaltons, right? And they're saying that, you know what, there's actually some, well, that's the 40, so the 45 kilodalton thing is the homodimer, but it's composed of two 22.5 kilodalton
that's
monomers.
not what it's saying. It's asking what kind of a change
Uh-huh.
increase the chances
Uh-huh.
for a 20.
Yep.
Like under what conditions would the monomer be 22.5?
Well, the monomer is already 22.5, but the dimer is what we have, that is the 45 that shows up on the SDS page, right? So they're asking, what can we do to make it so that we're breaking the dimer up into the two monomers, and each of the monomers are 22.5 kilodaltons.
We oxidized it in that problem, right?
Kind of, kind of. But yeah. So
And that make B wrong. Oh, but a reducing agent would oxidize it.
it would reduce it, and so
Huh?
so the disulfide bonds, right, can be broken if you reduce them.
Okay, yeah, we reduced it to the two um cysteine thiol.
Yep. Two of these, right?
Yes.
So they're asking us, so they're saying that this homodimer is 45 kilodaltons, and that's the homodimer. Um, but they're
Break it up.
telling us that the homodimer is made up of two of the same 22.5 kilodalton monomers. And if the SDS page, which is denaturing, right, gives us the homodimer, the only other thing we can do is reduce it to do a reducing SES page, which will break the disulfide bonds, and that can give us the 22.5 kilodalton monomer.
That's not removing the SDS, is it?
Oh, no, it's
It's B.
adding the reducing agent. Yep.
Okay. Because removing SDS, it wouldn't happen. The gel running time wouldn't do anything because it would get stuck where it gets stuck. And then a higher voltage, I don't think that they use voltage.
They do, just because they're passing an electric field, electric current through this. But yeah, that's not going to make it so that we're going to see the monomer. It's going to be, yeah, reducing it, because you have the native gel, you have SDS, which is just denaturing. You're breaking apart pretty much like all the non-covalent bonds that make a secondary tertiary structure. But it can break all that stuff except for disulfide bonds. For that, you need reducing conditions.
Okay.
Yep. So, okay. So tomorrow we're gonna meet around like 4, and
Said that was okay.
what's up?
She said that was okay.
Mhm.
Okay.
Yep. And so we'll talk about neuron stuff, depolarization. We'll talk about just some naming. We'll talk about Purines, Pyrimidines. We'll talk about uh protein structures, enzyme classes, and yeah, just stuff like that.
Okay.
But yeah, I think you're in a good position, and um yeah, we'll just kind of go over some of that stuff tomorrow, just to kind of, you know, we're not going to do any more questions like this because, you know, it's going to be more of like a chill session, but we'll just kind of talk about overall like topics and stuff.
All right. Sounds good.
Are you getting enough like sleep? Your test is what, at 8 a.m. on Saturday?
I'm sure they don't do any other times anymore.
Got it. So yeah, we'll end our session like early. And then I'll just have you go to bed. Like, it's hard for people to fall asleep the night before their test. So I usually tell them to go to bed like 2 hours in advance or something like that, just to make up for that. Um, but but yeah, so
All
awesome.
right. Good. All
All right,
right.
I'll see you tomorrow, okay? Take care.